Hi, everyone. My name is Momo Vuyisich. I'm a founding Chief Science Officer and Head of Clinical Research at Viome Life Sciences. And I will talk to you today about the platform that we've built called ViOS and how we're using it to identify human and microbial determinants of chronic diseases and cancers with the focus on RNA instead of what most people do, which is DNA. So let's get started here. I will tell you a little bit about Viome Life Sciences and this platform that we've built. So far in the last 6.5 years, we've recruited 16,000 or more clinical research participants. We have 300,000 customers in 106 countries. And all of those people who are associated with our platform have provided samples, clinical samples, stool, blood, saliva, urine, tissue biopsies, and so on. So we have more than 450,000 samples analyzed, and they're stored in our biobank. And they've all been analyzed with RNA sequencing so far. Therefore, we have generated more than 700 trillion RNA bases of sequence information from these samples. And with these samples come very rich metadata that I will discuss soon. And we are able then to do machine learning on all of these data to identify, like I said, molecular determinants of chronic diseases and cancers. And we have one mission at our company, which is to put chronic diseases and cancers in history books. That's what we work for every day. So ViOS is the systems biology platform that we've created starting from about 2010 when I was a scientist at Los Alamos National Laboratory. So the core technologies in terms of laboratory and bioinformatics were developed at Los Alamos National Lab. And then in 2016, we started the company. And this is still an open platform, which means that we have a grants program, and we basically would like to partner with many, many people around the world, individuals, organizations, whether they're physicians or scientists, and exploit this platform for generation of many other applications. But I will talk to you about the applications that we've generated so far and the biomarkers we've developed so far. So this is basically one of the takeaway slides from the whole presentation, which is the summary of the ViOS or the Viome Operating Systems or the systems biology approach. So we take lots and lots of people from all around the world. We collect their molecular data from clinical and sometimes environmental samples. We do collect, for some clinical research studies, environmental samples to look for fungi and mold in built-in environments. And then we collect comprehensive metadata, which may be clinically validated surveys that are PROs, patient-reported outcomes. They may be medical records, and they may be daily and weekly updates via the chatbot that's built into the app. For every chronic disease and cancer of interest, we perform machine learning on all of these data, molecular data and metadata, and the machines return these machine-learned models. And that's really what the rest of the talk will be about, is how do we utilize these machine-learned models as biomarkers of diseases for nutritional applications, for diagnostic applications, and the focus here is obviously precision diagnostics. And so the machine-learned model basically returns a list of features, which would be here in rows, and then their association level with that phenotype that you're measuring. And so these are all positively associated with the phenotype, meaning the more of this feature is associated with a higher phenotype, for example, a disease, whereas the blue features are anti-correlated. The higher the disease level, the lower the relative abundance of this feature. Now, the three features that we measure by default in every sample are the microbial taxonomy. So that is the strain level taxonomic classification of all microbes. That includes bacteria, archaea, fungi, parasites, viruses, phages, and so on. We measure the level of microbial gene expression in every sample, and we measure the expression level of all human genes in every sample. And so you would think that, for example, in stool, we would be only measuring the microbial gene expression, but that's actually incomplete. There are lots and lots, to the tune of billions of human cells that are shed every day inside the stool, inside the intestinal lumen, which we then capture as human transcripts. And these human transcripts can be highly valuable as biomarkers of diseases. So in saliva, we also see both human and microbial transcripts, and I'll discuss that a little bit more later. This is just a Viome operating system architecture. It basically has these 4 layers where in the first layer, we're performing sample collection for biobanking purposes and laboratory analyses. We're currently very focused on metatranscriptomics, but we are also adding some multiplexed immunoassays for whole blood, which we use for research only. They're not yet commercially available. And we perform some limited PCR, but any additional analyses can be added to this layer. We then have bioinformatics. I just discussed that we do lots of different kinds of bioinformatic analyses. And then we have application enablers. These are different tools that we use. And then finally, we spin out applications that I'm going to spend most of my time on. So one of the first things that people ask about, well, wait a minute, everyone is studying DNA. How come you're studying RNA? What is the value of RNA? So I wanted to spend a couple of minutes covering that because people who've been in the DNA world for so long, they forget sometimes the basics of the difference between RNA and DNA and why RNA may be actually a better, or at a minimum, a great complementary biomarker to DNA. So first of all, I think RNA is far more informative than DNA because DNA just means there's a potential, but it doesn't actually tell you what's going on right now. And I demonstrate that very clearly here that if you pick 3 tissues from one human, their DNA is going to be the same. However, their RNA is very different. And the reason their RNA is very different is because these 3 organs look very different and they perform vastly different functions. And that's all due to their RNA and not due to their DNA because their DNA is the same. In addition, when we go into chronic diseases and cancers, we can see that RNA can actually tell the difference between the intestinal lining of a person with Crohn's disease during remission and relapse in a very, very dramatic way, whereas their DNA during remission and relapse a month apart is identical, truly identical. And so RNA can not only distinguish different functions of different healthy organs, but it can distinguish between a healthy state and a diseased state. And those are very important factors in our choice of RNA as a new disease biomarker set. On the microbial side, we have very similar situation where you have many, many thousands of microorganisms called pathobionts. And pathobionts just means that they're commensal microorganisms that can turn pathogenic, but they are the same genetic background. They have the potential to be friendly and they have the potential to be pathogenic. And so if you sequence their DNA, it's only going to tell you the same thing, that it has the potential to be both things. But if you sequence RNA, you can actually tell the difference very, very nicely, whether they're expressing the virulence factors or not, and whether they're commensal and producing beneficial chemicals for us, or they're being pathogenic and producing virulence factors and causing us disease. And so it's very important that we look at the RNA profile of the microbes as well, and that's what we do in every one of our tests. In addition to this distinguishing the 2 states of a microbe, we can also look at the gene expression profile of the microbes. And you probably know that very few genes are expressed at the same time. Microbes don't express all the genes all the time because they basically adapt to their environment. And so it's very important to understand which genes are expressed at what level. And even though microbes have a leaky gene expression, they do express some absolute minute amounts of most of their genes at all times. Those don't really count as important in our physiology. It's really the most important, the most highly expressed genes. And we measure those very well with our metatranscriptomic approach. So in summary, I guess, this systems biology approach takes into account multiple locations of human cells, so blood, stool, saliva, and so on, but it also heavily takes into account the microbial content of all human tissues, especially oral cavity and intestines, because most of the genes expressed inside and on the human body are actually microbial, not human, and this has been well published and documented. So how do we collect samples in 106 countries? We have developed a method. This was all done before we started our company. We developed a sample preservation method for any human tissue that preserves RNA for 4 weeks at room temperature, and that allows, and we've developed these self-collection kits that allow anyone in the world to collect stool, blood, and saliva at home with just simple instructions, collect the samples, preserve them with these liquid preservative that we have, and then ship them just normal snail mail back to our labs anywhere in the world for analysis. And so these are the 3 sort of These are the three gold standard analyses that we do on everyone, whether they're a customer or a clinical research participant, even though not all customers get all three kits, but we try to do that to the best of our ability. And so stool is collected and preserved. Blood, we only collect currently 200 microliters of blood, but we're trying to scale down to 100 microliters of blood just via finger prick into these little mini vats. And then saliva is collected in a custom collection device. We use this device also for urine collection for clinical research. So metatranscriptomic tests that we currently perform are saliva, stool, and blood for paying customers, and they're using a universal laboratory method. So the robots are performing the exact same analysis. It's just full metatranscriptomic analysis. We're sequencing all RNAs. We're not selecting for any RNAs. We're sequencing all of them, whether they're microbial or human or from any other source. They're all clinically validated and CLIA licensed in the United States. All of them are unbiased, so we get to see all microorganisms. And I already discussed what kind of analyses we're performing: strain-level microbial taxonomy, gene expression of all microbes, and human gene expression. And this is all performed in a fully automated CLIA laboratory, so there's no human bias introduced into the process. For research and development, or for our very large clinical research programs, we also analyze a whole bunch of other sample types for different purposes, and these will eventually likely make it to the consumer and potentially make it to the diagnostic areas. But for now, we're just doing research. So we welcome anyone to apply for our grants program or partner with us, where we can explore the potential of RNA biomarkers in these sample types for various diseases and cancers. This is just a summary slide. You can look at that later if you'd like, but we think that metatranscriptomics is the best single omics method. It has huge advantages over the DNA-based methods for sequencing microbes, and that's listed in this table, in that we're seeing all microorganisms. We see them at the strain level. We see only the viable microorganisms, not just whether their DNA, remains in the sample. And we get to see both human and microbial gene expression levels. So those are all very big advantages. All of our methods for saliva, stool, and blood have been clinically licensed and peer-reviewed, published. So all of this is in publication, so I won't spend too much time here, but just highlighting that we've clinically validated the stability of RNA for 28 days at room temperature. We have shown very high accuracy of our entire platform at the species level. We have validated the validity of our finger prick blood whole transcriptome profiling relative to venous blood. And we have shown very high reproducibility. This particular data set is from our saliva testing for our cancer detection test. Lots more publications are forthcoming, but there are several others that are also published in addition to these. Here, this slide demonstrates the power of metatranscriptomic analysis. If you want to, for example, look at a solid tumor, you have the tumor cells, you have the infiltration of the immune system cells, and you have microbial components, which can be bacterial or fungal. And so if you want to look at them all in using different methods, then it's going to be very difficult to compare between samples the relative abundance of all these components. And so one unified method, such as metatranscriptomics, can actually provide relative abundances of all of these components in every single tissue sample. And so we've done this now analysis for several types of cancers, and we're actually going to submit a preprint for this in the coming weeks. But basically, metatranscriptomics enables one to look at both the cancer cell gene expression profile, the immune cell infiltration, and all of the microbial components, both in terms of taxonomy, so strain-level taxonomy, but also, very importantly, the expression of microbial genes inside the tumor microenvironment, which has been shown in the literature to be potentially aiding cancer in avoiding the immune surveillance. So our crown jewel application of this Viome operating system is this cancer detection test. We analyzed saliva samples from patients with oral and throat cancer and healthy controls. This was a collaboration with Queensland University of Technology. Like I said, we love to partner with other universities and biotech companies and large pharma in order to devise additional applications. And so with QUT, we partnered and we found out that saliva has very strong signals for oral and throat cancer. You can see here that our risk score that is computed for each person, you can see that it very nicely separates patients from healthy controls. And we did a very large study on 1,200 participants total and computed our specificity and sensitivity on an independent data set, submitted this to the FDA, and the FDA gave it a breakthrough device designation, which enables accelerated reviews for our device trial. And it also gives a much higher confidence in this test because there was a very thorough review of this application. So currently, we are offering this test as a direct-to-consumer test. We have a nice kit that anyone can purchase, and soon we're actually going to open it up to providers, so to dentists and doctors who would like to order this test, because based on the stage shifting mechanism or mathematical model that we built, we demonstrated that this test will actually save human lives because it can identify cancers or detect cancers in stages 1 and 2 that are much more treatable than stages 3 and 4. We can actually shift the detection stage from later to earlier stages and prevent much harder problem to solve in terms of treatment. I talked earlier about our metadata, and so everyone who is associated with Viome in either a research setting or as a customer is asked to complete a very extensive patient-reported outcomes survey that consists of 36 health surveys, and most of them are clinically validated. And so these obviously have go, no-go questions, so no one person will complete all of these. But based on go, no-go questions, we will drill down deeper and obtain highly accurate data on a very large population for all of these 36 indications. And so this is just a snapshot here. This is just a snapshot here of our current database and what kind of diseases we have in our database, people with what kind of diseases and what their numbers are. And so you can see here that we have many thousands of people with many different diseases with very rich metadata in terms of their indication that they're diagnosed with, what their current disease activity level is, what medications they've been taking, what supplements they've been taking, what diet they've been having, what other symptoms they've been having, what comorbidities they've been having, and so on. And so all of that is in our data lake where we access and do machine learning to identify biomarkers of various diseases. So currently, like I said, we've done quite a lot on this detection test for oral and throat cancer. It's on the market. And we're still proceeding with some more work, including the device trial for FDA clearance. But in the meantime, we're pursuing other opportunities as well. And so the next one is IBD. So we have very strong data on IBD. You can see here that we have 1,067 people with IBD. We have very rich data set in terms of their medications and comorbidities that we can match them to 10,000 people who do not have IBD. And then we perform machine learning on that. This is just one sliver of a data output that we obtained from these large machine learning studies, where we're looking at heat maps of human signature. This is basically referring to the level of human gene expression is plotted in the rows of specific human genes that are associated with that disease. And then the columns are in this space, in this particular graph, are the microbial species that are also either associated or anti-associated with the disease. And these correlations are all plotted in a heat map plot. And so we plot a lot of these graphs and make very good model outputs. And so this is one of the outputs of the model for IBD. And so what you can see here is that we have some very good biomarkers of IBD. And I'm going to focus here in this particular box here, which these biomarkers here have the highest differential in gene expression level and the lowest p-value for association with IBD. And what you can see here is that we're seeing TNF-alpha as a biomarker as well, but it's not nearly as good as this one, even though this is sort of the gold standard today. And then we're also seeing the calprotectin diagnostic reference here. So we do see calprotectin transcript in our data, but it's a very poor marker relative to these best markers that we have. And so these biomarkers now can be used for either diagnostic purposes or therapeutic purposes because these are very strongly associated with IBD. And so our current IBD model has ROC AUC of 0.83. We're still refining this model. We're still gathering more data from clinical research participants, and we're probably going to do an independent validation sometime this year, and then hopefully bring it to the market. We are currently trying to develop a model that distinguishes IBD from IBS patients. That's our near-term goal. We've also worked very hard on type 2 diabetes. So we've been able to recruit more than 50,000 clinical research participants from our customer database and develop a very precise, well, very accurate type 2 diabetes risk model. And so this risk model takes into account the human and microbial features. And these are the values for this risk score for people who are non-diabetic, pre-diabetic, and diabetic. And we have very low p-values here. So in summary, we have a huge database of molecular features and metadata. We can do a lot of machine learning. We're looking for partners. Here's a list of our peer-reviewed publications. Here's a list of our preprints that are, most of them are under review. And we're happy to partner. Please contact me if you would like to partner to develop new applications in this space. We are in Seattle, where I am, and we also have an office in New York City for our computational biologists. And please contact me, and hope you enjoyed that.